Welcome back to Acing AP Biology. In this lesson, we will go over the specific types of receptors and signal transduction pathways. G protein-linked receptors are the most common type of receptor. It requires the input of one ATP molecule. A ligand binds to the G protein-coupled receptor and it becomes activated, changing conformation. This leads to the phosphorylation of GDP to GTP, which activates the G protein. The activated G protein then binds to the inactive enzyme and activates it. The activated enzyme initiates a cellular response. Another type of receptor is a ligand-gated ion channel. When the ligand binds to a specific ion channel receptor, the gate of the ion channel opens, allowing for the rapid flow of specific ions into the cell. This spurs a cellular response. Once the ligand is released, the gate closes. These are important for neurotransmitters. A specific type of transduction pathway is the phosphorylation cascade. The ligand binding to the receptor is the first step in a signal transduction pathway. The following steps amplify the signal, converting the information into different forms until the response is generated. Relay proteins pass on the signal to different molecules. The protein kinase is commonly used in signal transduction pathways as they catalyze the phosphorylation process involving the transfer of a phosphate group from an ATP molecule to a protein to activate it. Phosphorylation induces the activation of target proteins. This is called a phosphorylation cascade. Dephosphorylation removes the phosphate from the activated protein, reforming ATP, and this inactivates the protein, which stops the signal transduction pathway. Dephosphorylation is catalyzed by protein phosphatases. A ligand binds to the receptor, activating a relay molecule. This relay molecule activates protein kinase 1 by initiating phosphorylation. The activation of protein kinase 1 leads to the activation of protein kinase 2 after ATP is phosphorylized. Protein kinase 2 activates another protein through phosphorylation, which then leads to a cellular response. After the cellular response, protein phosphatases initiate dephosphorylation by removing the inorganic phosphate groups from the proteins, deactivating them, and stopping the cascade. Signal transduction can also take place through second messengers. Second messengers are non-proteins that act as relay molecules in the signal transduction pathway. The most common types are cyclic AMP and calcium 2+. The pathway involving cyclic AMP, also known as CAMP, starts with the ligand binding to the receptor on the G protein-coupled receptor, activating the G protein. The G protein then binds to adenyl cyclase, which is an enzyme. The activation of this enzyme leads to the activation of CAMP through the phosphorylation of ATP. CAMP then activates a protein kinase molecule, initiating a phosphorylation cascade and eventually a cellular response. Normally, cellular responses involve the activation of enzymes or the turning on or off of genes. usually involving transcription factors. That's it for Unit 5. See you next time. For more bio content, hit subscribe.